My name is Dr. Laura Lee Johnson. I'm the Associate Director of the Division of Biometrics 3 at the Food and Drug Administration. I'm also co-director of this course, so thank you for coming. If you have any electronic devices on and you're in here, please go ahead and turn those to silent mode. Thank you. So standard disclaimer, and the reason I put the lecture notes on effect modification and confounding at the end of the last lecture I gave is because it always bothers me when people only cover this in epidemiology and when they're talking about observational studies. So we're going to cover them a little bit tonight, but keep in mind that these are problems whether you have a randomized or non-randomized study. So if you talk to a statistician, they're going to kind of break your variables up into a handful of different types. Effect modifiers, potential confounders, and other things, so other covariates. And realistically, we tend to call everything in the model, all these X's that you'll eventually learn to love by December or so, we call them covariates. So effect modification is when you have an interaction. Some folks call this synergy. The trick here is that it could be larger or smaller. What you're interested in is the association between the outcome variable and some other variable, in particular the intervention, is modified by different levels of a third variable. So what do I mean by that? When I'm talking about that here, I'm thinking about something like, let's say I have a new cardiovascular drug. And if I give that drug to men or to women, it's going to behave differently in changing their blood pressure. That's effect modification. A classic example is when we look at smoking, asbestos, and lung cancer. So smoking alone increases your risk of lung cancer by a certain amount, we'll say by an amount A. Asbestos exposure alone increases your risk of lung cancer by some amount B. The question that came up was then if you were a smoker and you had asbestos exposure, was your risk of lung cancer A plus B? Or is it some amount that's higher, so because you have that asbestos exposure, maybe it makes the smoking that you're more likely to get lung cancer? Or in fact, maybe having asbestos exposure is protective, so those smokers are in fact less likely to get lung cancer. This is the idea behind effect modification. Now, instead of actually going through a huge amount of definitions, Neil Pierce has a beautiful chapter. So if you do your favorite search engine and type in a short introduction to epidemiology Neil Pierce chapter, you will find about 22 different slides where he goes through what is effect modification, defined for statisticians, a different definition and set of examples for public health workers, for lawyers, biologists, lots of different professions. So please read Neil's chapter, go through it, and find the definition that fits your profession and see how that works for you. And I'm going to move on to confounding. So confounding is when I have two or more variables. And the trick that we're going to keep talking about tonight, and that some of us already started talking about on the discussion boards for the last lecture, is they may be known or unknown to the researchers. So one of the common themes through tonight's lecture is there's a lot of information you know. If you know it, you might be able to collect data on it. Not always, but maybe you can. If it's unknown, you're probably not collecting that data. That's your biggest worry, is what you don't know. So these two variables, or additional variables, are confounded when their effects on that common response variable or outcome are mixed together. That means I'm incorrectly estimating the association between my exposure and my outcome because I haven't taken into account a third factor. So my friend Pam Shaw came up with a really simple example. It may seem silly, but just like these different vans that I use in another example, it helps me remember the concept. Consider if you saw an association between carrying matches in your pocket and lung cancer. Are the matches causing the lung cancer? Or is it the association between carrying matches and lung cancer probably some unmeasured variable associated with both, like cigarette smoking. So you have the matches are actually to light the cigarette. The matches aren't causing the cancer. It's smoking the cigarette that's causing the cancer. This is the issue with confounding. If you never measured their smoking status, but you measured the matches, you've got the wrong actor. An example that was in the news for many years, it was in the New York Times in 1981. Study links coffee use to pancreatic cancer. So we have very few ways to treat pancreatic cancer. Even the methods we have, especially for adenocarcinoma of the pancreas, they don't increase your lifespan to a very long period of time. So this came out of a New England Journal article in 1981, and they were trying to figure out what were some of the causes. So pancreatic cancer probably doesn't cause you to start drinking coffee. Therefore, we think this direction of the arrow is probably correct. Coffee drinking might cause pancreatic cancer. The problem is coffee drinking and smoking tend to go along with each other. Now, I drink coffee, I don't smoke. But this doesn't have to be a perfect association. You just have to have some association. So what's the confounding example? There's a relationship between coffee and pancreatic cancer, but smoking's the known risk factor for pancreatic cancer here. Smoking's associated with coffee drinking and vice versa, and it's not directional, so there's not like smoking's a result of coffee drinking. You're not in a line. In fact, the picture looks something like this. So I know I have a direction between my smoking and pancreatic cancer. Smoking leads to pancreatic cancer, coffee drinking, leads to pancreatic cancer, but there's no other directionality here. So what's confounding? If an association is observed between coffee drinking and pancreatic cancer, the coffee actually causes the pancreatic cancer, which is this, or the association is a result of confounding by cigarette smoking. So I'm measuring the coffee. I'm not doing a good job of measuring the smoking, but this is basically the relationship I'm looking at. And in fact, after many years, the decision was this was the answer. Coffee drinking has nothing to do with your pancreatic cancer. So how do you handle confounding? Well, the way you do it is you identify all potential confounders you can think of. You measure them. You're going to probably measure them in many different ways. How do you measure smoking? Current, former, never? How many pack years you have? There are tons of different ways to measure smoking. In the data analysis, you might decide to use stratification. Although what most statisticians will tell you to do is don't stratify, smoking status. Instead, stratify, or don't stratify. You go ahead and put that variable in your model, which is sometimes called an adjustment. So I'm going to put a covariate in the model. We'll talk more about that over the coming weeks. But your biggest problem here is fearing the unknown. Now, I will also say that the Harvard colleagues who worked in that 1981 original article on pancreatic cancer actually did try to control for cigarette use. But part of it was the sampling of their cases and controls in the study and the categorization of coffee, which we'll talk more about tonight. This next issue comes up in randomized trials as well as observational trials, and that is residual confounding. This is when you have that poor measure of a confounder. For example, how do you measure physical activity? Even when we put a confounder in the model, so I put some measure of physical activity, maybe it's how many steps people walk on average per day, some other method, I'm not really explaining the full effect of physical activity in the model. Again, smoking is another example. What variable are you putting in? And is it the correct way to actually model smoking inside that entire relationship? Randomization helps protect against confounding. It does not prevent confounding. When Dr. Joachim comes next week and talks about randomization and hypothesis testing, part of what he's going to talk about is, what randomization gains you. So if you don't randomize, or if you randomize poorly, so you don't actually follow the process you've set up, the problems that can result. But this is kind of like buying insurance in a way. It doesn't mean something bad won't happen. You're just trying to prevent it as much as possible. You can have non-random dropout or attrition in different study arms in a randomized study. You have to have a way to account for that. But what randomization tells you is that for all of those known and unknown confounders, something is by chance, or you're able to dig down and figure out what's possibly causing it. You know, a big problem in some of the early HIV trials, even sometimes today, and a problem in a lot of the trials I used to work on, was that patients would test the substance. And they would drop out if they weren't getting the drug that they wanted. So you'd try to do a blinded trial, and they would essentially go to a compounding pharmacy or someplace else to have the substance tested. Or in one of my studies, they said, well, I am going to go buy more of this substance because it was available in a lot of different stores. They went and bought a lot of isoflavones and took it because they wanted to make sure they were getting the isoflavones. Someone's shaking their head no, and that's right, don't do that. But that's also part of our consenting process. Someone who's going to do that you hope is not in the study. But at the same time, when you're dealing with people who are desperately afraid they're going to die, such as in some of those early HIV studies and a lot of other studies, you have to figure out how you're going to balance this information and how you're going to handle it in the analysis. So this is a picture from John Power's lectures. And he talks a little bit about confounding and effect modification sometimes. And this is a nice little pictorial example where you have the outcome. All right, now I have to balance what I have here. All right, the outcome is kind of your y-axis. If you have no confounding and no effect modification, across the baseline variable values, you're going to have standard effect size, the test and control arms are going to look the same. This is what you hope for. If you have confounding but no effect modification, as that baseline variable, for example, may be increasing, the impact you would expect to see on the outcome is increasing, but the distance between the test and control arms is the same. So kind of the impact is the same regardless of which study arm they're in, but the baseline variable looks differential. If you have effect modification is when the slopes of the lines look different. And if you have confounding and effect modification, it's going all over the place, right? Both have a slope, but they have different slopes. So this is what I spend my time playing with on Saturday nights, right? It's trying to figure out in my placebo groups, how are they behaving? And then in my test groups, how are they behaving? And is it due to anything else? Or is it due to the actual intervention? So moving on, tonight we're going to talk a little bit more about epidemiology. By the end of tonight, hopefully you can identify some of the common observational study designs and understand why they may or may not be used to answer a research question. So in the lecture after this, you're going to hear from two different people. One is Jerry Sachs, and he's going to talk about clinical research from a patient's perspective. An important element to talk to patients about is what type of study design are they willing to be in. There are some people who are willing to be intervened upon. There are some people who are willing to be randomized. But there are other folks who don't want to participate in those studies, but may be willing to participate in something like a natural history study, or may be willing to give some of their data for observational research in general. Study participant selection is also going to be talked about in the next lecture by Catherine Stoney. So Kate Stoney's been involved in all sorts of different types of studies, so make sure to ask her tons of questions. Also because she's a friend of mine, so it's always good to ask her. But when you get to next week and the overview of hypothesis testing, I do want you to consider that most of what you're going to learn about hypothesis testing is for randomized trials. That doesn't mean it doesn't apply to these observational or non-randomized trials. So Dr. Joachim is going to try to give some additional examples that are non-randomized, but you can also ask him questions about these types of studies. And I want to give some thanks to Jerry Menikoff at OHRP. He'll be talking to you about institutional review boards because he helped rearrange and edit some of these slides about a year or two ago. So what are our definitions? What is this epidemiology that I keep talking about? There's a sign that I saw when I walked in the building today. It was like, why me, why now? That's kind of what you figure out in epidemiology. So epidemiology is the study of the distribution and determinants of disease and injury, whether it's human, animal, plant, or any other population. Veterinarians use epidemiology. Folks that are working in forest services use epidemiology. Everyone uses epidemiology. The basic tenets is that disease does not occur at random, and that there is some type of causal and preventative factors that can be identified if we do a systematic investigation of different populations or comparing subgroups of individuals within a population. Now, what most people think of when they think epi, which is what people shorten epidemiology to, is they think epidemic. So there's a popular book several years ago called The Hot Zone. Interestingly enough, Ebola is back on our minds again. And then there were movies and all these others. So you see these TV shows and such. It's far more exciting than what most of my epidemiology friends do. Most of them sit in front of a computer, or even the ones in this service that goes and handles outbreaks. They go and they're on a lot of cruise ships because people get something called the norovirus. But epidemiology is more than just studying these epidemics. It's also these big cohort studies. So the nurses' health study, for example, and the cardiovascular health study. There's actually a big cohort study that several of your classmates started talking about online, looking at Ebola survivors, trying to do a large long-term natural history study. There are many things in between. But epidemiology is really considered by some to be the cornerstone of public health research. One of the articles that you all have for tonight's lecture is a New York Times Magazine article on epidemiology. And I encourage you to read it because it's written for people who read the newspaper. It's a pretty nice, common sense article that I think explains the basis of why we actually want to use epidemiology very well without overselling or underselling the shortcomings. So are all these big studies refuted? There were a lot of epi studies prior to the Women's Health Initiative. So the Women's Health Initiative was a large, it was kind of a joke to some folks that women had not been studied in most NIH trials, and then they tried to do one trial to answer all questions that had never been addressed. That's not exactly what happened, but it almost felt like that. But this was a huge study to look at both some epidemiologic aspects and some randomized trial aspects. If we did hormone therapy, hormone replacement therapy, what were the impacts on bones, on cancer, on cardiovascular health? There had been a lot of epidemiologic studies, and people said we already knew the answer. But realistically, those studies did not agree. And in fact, after the Women's Health Initiative was stopped early in their randomized trials because it was seen that, in fact, there were not many positive benefits for most of the outcomes. There were for some, but not for all. By then, we had stronger computing power and new statistical methods. And with the knowledge we had, Ross Prentice went back and actually reanalyzed almost all the epi data that had been used to justify WHI and said, actually, we can reproduce the randomized studies. Now, that helped give credence to that, but I'll tell you the number one thing that was missing from those analyses was socioeconomic status. So that's the problem, again, when you're doing non-randomized trials. You don't always know all the variables you should be putting in the model. Now, prevention is something that we would love to study with randomized studies, but it's very hard for me to tell people to actually follow a certain lifestyle for 20, 50 years and wait for the outcome. Prevention's a huge place where we use epidemiology. So this is the link to New York Times Magazine, or you have a PDF of the article if you want to look at it. The sum is that epi is hard. You've got to measure a ton of things. You've got to measure it in a lot of different ways. And you have to do it very accurately, fairly often, because a lot of these variables are changing over time. You want to not lose any data, so you've got to track everybody down at every single time point. You've got to measure it the same way every time. And this is a problem because our methods of measurement may be changing. Also, where we make diagnoses may be changing, so you've got to work all of this into your protocol and analyses. But you're never going to know what you don't measure. I do like the phrase that I forget who used this, that epidemiology is like hypothesis-generating evidence. This is kind of like circumstantial evidence in court. Or like watching some of those TV shows where it's like, you're like, I know who did it, it was really clear, but they've got to actually prove it methodologically. Epi is that circumstantial evidence. But it also might be your only evidence outside a laboratory. There are times that I can randomize patients, but I can't randomize you to smoke or not smoke. I can randomize an animal in the lab to be exposed to cigarette smoke, yes or no, but I can't randomize humans. So there are still, as we talked about last week, fundamental limitations on these observational studies. I can distinguish an association, but I can't inherently determine causality unless I figure out another way. But how else am I going to generate hypotheses? So I love epidemiology. I think it's hugely challenging, but I think it's a real cornerstone and important. But it's especially important for generating hypotheses because you've got epi, you've got clinician experience and observation, which realistically, when well done, is epidemiology, or they're pulling it out of thin air. I'll take either of those first two. Thank you very much. So I'm going to mention a lot about cancer and smoking tonight because there's a lot of information there. But it was in the discussion of if smoking was tied to lung cancer that we came up with a criteria of how you actually do causal inference from observational studies. So this group of folks set up a list of epidemiologic criteria for that. You've got to have statistical significance. It's really easy to get statistical significance. Paul will talk to you about that in the hypothesis testing lecture. And I say that, and everyone who's never gotten it was going to complain, but like, if you have a high enough sample size, you'll find statistical significance. May not be clinically meaningful, but you'll have statistical significance. That gets me to the next one, the strength of the association. You want to have consistent strength of association that's meaningful. Now, whether that's in an odds ratio, relative risk, et cetera, doesn't particularly matter. You want to see a dose-response relationship. One thing to consider is it might be a U or an upside-down U, but typically, as I increase your exposure, you should be having increased responses. There should be a temporal sequence. The cause has to happen before the effect. You would be shocked how many times that's where it falls down for the causal inference. You have to have this consistency of the association and replication of the results. And Dr. Stoney will talk more about this in her lecture. You need biological plausibility. True story. I sent an explanation of some numbers to a PI, and I had run the wrong model, it turns out. So I figured this out. So he's like, oh, this is great. He sends me, when I first sent the numbers, he sends me this whole reason why this makes sense. I realize I've made a mistake. I'm like, actually, it's the inverse of the numbers that I sent you. Oh, that's fine, and gives me a whole other explanation for it. We don't always know, and he was a totally good investigator. Like, the thing is that we don't know enough about the biology. A lot of times we can make a very plausible story going in almost any direction. So while it should be biologically plausible, and maybe we just don't know enough yet, or we can still make the story work. But the last one, this H, is a key. Experimental evidence. What does that mean? Maybe you're randomizing pigs in the lab. Maybe you're randomizing rats or mice. Somewhere, somehow, you in fact did run the experiment. It just may not have been in human beings or in the animal or the plant or whatever that you want to actually run the experiment in. Maybe it's not in the forest, it's in the lab. But somewhere, somehow, there has been experimentation such that you're able to show that there is evidence of what's going on beyond the epi. So this isn't necessarily going to be as strong as an interventional trial, but you're going to hope that it's better than nothing. So this gets to this experimental versus observational element. Epidemiologists use lots of different types of studies. So tonight, I think I had labeled my lecture just observational studies, and I noticed it said epi. As a shout out to all my friends at the Center for CDC, why am I completely blanking? But it is both also prevention at the end. There's a P that's not in their name. But the issue is they actually do a lot of interventional studies in epi. They tend to not be randomized, but it depends. But really what they're known for classically are the observational studies. But this is the one slide as an ode to what the CDC has, and they're very extensive online classes about epidemiology if you want to take them. Remember, the interventional or experimental studies are when the researcher influences what happens to the subjects. There's a very different concept of human subject protections there than when you do an observational study. And that is true in almost every single country. You still have human subject protections issues in observational studies. If you're in the United States, you're still going to have to go to an institutional review board or some group like that. And again, true in almost every country. But observational studies are descriptive. You're not intervening on people. That doesn't mean that by asking them questions or taking samples, something couldn't happen to them. But in these observational studies, you're kind of like a reporter. You're trying to gather information, the who, the what, the where, the when. And then on the analytical side, you're trying to figure out how and why. So now we're going to go through some of the study designs. Case reports and case series are fairly similar to each other. This is when, and this is why I was talking about, the physicians have a gut feeling. Well, if they're recording the evidence, case reports and series. Observations of patients with some defined clinical characteristic. It might be they have a disease, but it seems to be more severe, or they don't seem to be part of the population that normally gets this disease. Could be, in fact, that you just see a cluster of symptoms that seem odd. A case report is going to have a description of data, typically without a comparison group. And again, though, you need a well-defined population of people. You're going to hear me say that about 20 times tonight. Why does it need to be well-defined? Well, you need to have clear definitions of your phenomena because other people are now going to read this report, read about this series of cases that you saw, and try to see if they have that same group. So you need to be clear about what you know and what you don't know because you're trying to really define a disease. So what are the definitions that put individuals in this series and not in this series? That's what you have to define and make clear so other people can replicate that with their own patients. The observations you make need to be reliable and reproducible. Again, somebody else is going to go out and try to say, hey, do my patients have that blood level? Hey, do my patients have this characteristic? Done well, these are extremely useful. So what type of math do I tend to see? I tend to see means and medians, standard deviation of the error. I'll be honest, a lot of times people just publish all the data. So you'll see this table with information for subject 1 through subject 20. And it's here's their age, here's their sex, here's all this other stuff. Because if you have a small number, so if you're under, say, a sample size of 10, standard deviations and standard errors, while you can calculate them, are meaningless. But you may see proportions. Do you only have one female? Well, that's pretty important if you've got 20 folks. Stuff like that you're interested in. Should have moved the confidence limits and intervals up by the standard deviation. Same rule applies. If your sample size is under 10 or 15, that number's pretty not very reliable. We've also got separate data for subgroups sometimes. So sometimes they'll break it out like rural versus urban, men versus women, younger versus older folks. So they seem to have the same phenomena. So think about a really bad influenza. It might impact the very young and the very old. So I may say, okay, I've got these two different groups. I may put them next to each other on the table. But what may be really the odd characteristic is suddenly the only people dying from influenza are in their 30s and were relatively healthy to begin with. That's the type of thing that you put into a case series, and also characteristics that are then defining your group. So what do we use these things for? We've got hypothesis formation, we've got natural history. So the beginning of natural history studies are many times these case series or case reports. This is basically defining your clinical experience taking care of these patients. Do you have biased patient selection? Yes. In almost every case series that's published, it's a set of patients that like one group saw. It might be just a blip. But the idea is to figure out the generalizability of these results. If it is something that is in fact suddenly generalizable to larger sets of clinicians around the world, we may have an actual epidemic on our hands. It could be chance, it could be a characteristic. At this point, we don't know, which is why we're trying to record everything and figure it out. It's only with very careful measurement and recording that we can actually get this work done well. An example of where it was done very well, again from 1981, was the initial report of 5 cases of pneumocystic pneumonia in previously healthy homosexual men. First time the Centers for Disease Control and Prevention put sexual orientation in the discussion, let alone in the title of something in Morbidity Mortality Weekly's report. And actually, this was noticed here at the NIH also. But where this report started from was a pharmacist. At the time, this type of pneumonia was typically seen in advanced cancer patients. In order to get the drug in the United States, it had to go through a central pharmacy, and that lady, literally one person in charge of filling all of these, started noticing that the patients that she was getting the request for this drug for were very different than the patients that she had always given the drug to. And they started an investigation. And when they did that, they found out that, yes, the patients, in fact, had this type of pneumonia. They needed to be treated. But something else was impairing their immune system. It was not their cancer treatments. And that's what led to this report, and then additional reports that started coming in from across the United States and across the world that people were also seeing folks that matched these characteristics, and they eventually came up with a better definition. So, never underestimate in your clinical practice, and that clinical practice involves all types of clinical work, what you might see and how impactful that can be. So whether it's H1N1, whether it's the next Ebola outbreak, or whatever is going to come, because something will come, this work is important. So now we're going to get to cross-sectional surveys. Here's where we're going to observe the prevalence and characteristics of disease. Cross-sectional, remember, is the snapshot in time. Again, my idea is I need to understand the characteristics of participants in my study in a well-defined population. First things first, define your population. Am I going to look at all adults 18 and older in the United States? Am I going to try to find everybody that has a very specific rare disease around the world? Am I going to try to find a sample of people with this disease on the African continent? What am I trying to do? Define your population. Derive a sample of that population. who are you going to actually ask and give this survey to? It's probably not everybody, but then you have to make sure, do I have representativeness in who I'm giving the survey to? If I only give the survey to men, I can't really talk about women. If I only give it to 3 women, I probably still can't talk about women. Have I covered the age ranges, the different cultural issues that could be coming up, and a lot of other issues? If you're getting NIH funding, you have to report on your sampling about race, ethnicity, and sex. You'll get a whole lecture on that. You've got to define the characteristics that are being studied. What are the observations you're making? What are the questions and the responses to the survey? And what are the instructions? If you say you're measuring blood pressure or weight, how are you doing that? Are you taking a specific scale to people's houses, making sure that it's been properly adjusted, is going to measure correctly? Are you going to do it on the floor? Are you going to do it on carpet? How is that going to change your result? Are you just going to ask people how much they weigh? Are you going to do it in kilograms or in pounds? How many decimal places are you going out? Or are you not? You must have standardized observations. You must train the people making those observations on how to do it, and that's your way to reduce the measurement error. If you're asking for self-report, that's okay, but you have to acknowledge that when you're doing your study design. You need to have very clearly defined characteristics. Remember, sleep. I need to know more, not just how do they sleep. You need to have very defined questions. When do you go to bed? How long have you slept? How was that measured? And those methods of data collection have to be applied equally to all of your study participants. So if you have a question that you can ask people who are sick, but not ask people who are well, that's a problem. When I'm trying to do a survey, I need to figure out how I get the information from everybody. If I have somebody who's illiterate, who can't read, and I hand them a sheet of paper to fill out, that's a problem. So if I can't do the same method of data collection, I need to then be able to know that my multiple methods of data collection will not bias the results. And that's a big issue. So what about prevalence and incidence? I mentioned prevalence. So prevalence is, in a very simple term, the number of people with disease divided by the number of people at risk. Again, it's like taking a snapshot. If I asked how many diabetics, we'll say type 2 diabetics are in Brazil right now, that's prevalence. Prevalence is basically the incidence times duration. So what is incidence? Incidence is your number of new cases of a disease over some period of time, divided by the number of people who are at risk at that period. So if I already have type 2 diabetes, I'm not at risk of getting it again. So an example that you might ask is how many new incident cases of type 2 diabetes were diagnosed in 2007 divided by the people who could develop the disease in 2007. So what's going to be described in tables? When you're looking at these cross-sectional surveys, you get a lot of descriptive questions. You want to basically tell folks how common are each of these different characteristics or factors. What's the distribution of the factors of interest? Like, think about age. Are you more likely to get this when you're young, when you're old, somewhere in between? That's what gets described in the tables and discussing these surveys. You also are looking for these associations. The problem is we tend to look at them in 2x2 tables, but we're looking at associations. What's the relationship between the factors? And how do people that have one factor differ from those without? So this gets into that idea, again, of confounding and effect modification. So I might have some percent. They might talk about the prevalence. Sometimes they give you some number like 65 cases per 10,000 life years or something like that. Like, you'll see different numbers, or 10,000 people. The life years tends to be more with the incidence. Mean or median levels of relative factors, and subset the descriptive information by important subgroups. So if it's HIV, you may do something different for intravenous drug users, non-drug users. People have different sexual behaviors. You might do different tables. For the analyses, you tend to see a lot of categorical analyses. You want to use regression. Thankfully, the journals are getting pickier about this, but used to be I saw all these chi-square tests. So it's basically like, oh, what's the relationship between their body mass index category and their sex? What's like they would compare all these two variables at each given time instead of really writing a regression. So you want folks to use logistic or ordinal regression many times for their categorical variables, so that way I can add in all those potential confounders and effect modifiers and see the big picture for folks. If you have a continuous outcome variable or variable that you're looking at, you might do t-tests, but again, you should probably be using some form of regression instead. So just remember, if all else fails, regression is your friend, some form of regression. But since everybody uses 2x2 tables, I figure I should teach it to you a little bit. You've got this representative sample, and you can classify them. You know, we say outcome, but again, we don't know that there's a direct causal relationship. But let's say people had a disease or didn't have a disease, and then they did or didn't have a risk factor. Think back to John Gallen's talk, and I think he talked to you about that classic example with the wells. And you know, you could say, did you get your water from this well, yes or no? Did I get this lovely disease, yes or no? Classify people in these 4 different ways. There's actually this study in BMJ that was out of Africa looking at the efficiency of the BCG vaccination and preventing TB. Well, the nice thing about that vaccination is it leaves a mark on your arm, or the old version did. The new version doesn't, so you couldn't do it now. But you had a mark. So they basically looked and they said, okay, we're going to take a sample of people. Did they have the vaccination? Do they have the mark from the vaccination? Yes, no. And if not, then if they didn't, then they looked to see if they'd had TB, yes, no. So they titered out on that. So what's your positive attribute? The nice thing about these cross-sectional surveys is they're cheap if you have a common disease. So if lots of people are going to have the disease, I could probably figure out a lot of information about it. It's like a common influenza, common colds, things like that. You can get a lot of information very inexpensively. It is more representative of cases than that case series we were talking about. Why? Because I'm taking a broader population typically. They tend to be short in duration. A lot of these last less than a year. You have a very specific population that you're looking at, and you have simultaneous wide variety of measurements. The negative. If you have a rare disease, you might not pick it up. So I interview 50,000 people. If 1 in a million have this disease, I'm probably not going to have 4 or 5 people in my study, If it's unsuitable for a disease of short duration. So I mentioned the common cold, but think about it. How long does that cold last? Maybe 5 days or something? Are you going to remember it when I ask you if you've had a cold in the last year and it's been 8 months? Probably not. You're probably not going to remember all that much about how you felt. You also tend to get high refusal rates. And when you get high refusal rates, you get inaccurate prevalence estimates. So, do you want to fill out that 8-page survey? I'll tell you, in a lot of countries, this type of survey work that they do, if they're doing it for their public health, they actually make it mandatory. They'll throw you in jail if you don't answer the survey. Not all places. It's more expensive and time-consuming than a case control study, which we're going to discuss next. And time is our best and worst confounder. So trying to remember information can be questionable. So some examples where this was pretty good and where there was some interesting math. We had this study on the prevalence of overweight and obesity in U.S. children, adolescents, and adults. This was published out of the Center for Disease Control and Prevention. using something called National Health and Nutrition Examination, or NHANES data. So NHANES is a study that's launched every year by the CDC. They actually go in person, and they make you get on a scale, and they take your weight, and they take your blood pressure. So an interesting other study came out of CDC and the National Cancer Institute. Cause-specific excess deaths associated with underweight, overweight, and obesity. So most people will think, well, if you're overweight or obese, that's tied to higher mortality. The problem was the analyses that have been done left out possibly the most important thing that's tied to mortality, and that's your age. Actually, when you look at mortality, typically you want age and age squared in the model. And so back in the 80s, a lot of work had been done. They said, BMI around 28 is pretty good if you're thinking about mortality. Now, you need to take into account smoking and all these other factors. But then this huge landmark case went out. I will not lie, got our department a lot of money. But these folks were going through the analysis and the model, and they're like, where's age? Well, we don't have age in the model. Well, age is the best predictor of mortality. You've got to put it in your model. Once they put age in the model, you got very different results for cause-specific excess deaths. And so Kathy Flagel here, along with these other authors, actually had a series of papers saying, well, if you come down to it, and it went through extreme scrutiny from both the CDC and the NCI before it got published, that, about a BMI around 28 is better. So if you have normal weight, or rather, I should say, overweight can be protective. And there are a lot of reasons people thought that might be, but actually most of them were refuted by that data also. So it's a very interesting read if you want to look at that and then all the commentary that came afterwards. So don't forget to put variables in your model. But like age, sex, these are easy pickings, folks. Those should be in pretty much every model. Now, case control studies. Actually, let me back up from what I just said, which is sometimes it doesn't matter. So everybody thinks sex matters, and you look at it, you put it in the model, you take it out of the model, and nothing changes, or age squared. Usually these are variables you should always collect and always evaluate in your models, but in fact, it may not matter. So you have to let the data tell you the story. Case control studies. It's kind of exactly what the title sounds like. You have these observations regarding possible associations between a single outcome, typically the disease, aka what defines caseness, and one or more hypothesized risk factors or exposures. When you have a rare disease, this is where you start. Here are natural history studies, but typically you're doing a case control study. If you have diseases with long latency periods, you also tend to see case control studies. They are quicker and less expensive than the cohort studies we're going to describe after this. So a case control study, you're going to compare the prevalence or level of a possible risk factor between representative group of disease subjects, so your cases, and disease-free subjects, so your controls. They're going to hopefully come from the general same population. Why is that? Well, because I'm trying to control for the socioeconomic factors, all this other stuff that could be going on. But again, what you're never really sure of is what's the risk factor. So you have to be careful what you're controlling and that you're not over-controlling in studies. And a lot of case control studies will see multiple control groups, in fact, to try to tease this out. But the word representative is really key here. You need to make sure that your cases and your controls, in particular your cases, represent the disease under study. This can be hard because it's easier to recruit where you work, right? But realistically, the cases are all over the place, and you need to make sure that the factors you're teasing out aren't just due to location. So this is a simple picture. You're going to define everybody by their disease status. Disease, no disease. That means you have to be able to define disease and no disease. You have to make sure your no disease folks, in fact, have no disease. They may not know that they, in fact, have a disease. So you've got to be careful about that. But then we're going to try to figure out is who was exposed, who wasn't exposed in the disease group, who had certain exposures and didn't have them in the non-disease group. A lot of times, it's going to look exactly the same all the way across. Again, these cases are supposed to represent all patients who develop disease. Your controls should represent a general, I'm going to call them a healthy population. They may not be all that healthy, but kind of non-diseased again, population. And the information has to be collected in the same way. If I have people who have cervical dysplasia, and I'm going to go take a biopsy of that, those are my cases. The people who don't have dysplasia, am I allowed to take a biopsy? Where do I take the biopsy? You've got to think about how you're collecting this information. It's also hard because sometimes you have interviewers, and I think Barbara Stussman will talk about this some too. You'll have these interviewers and they have an idea, like, okay, the cases tend to have headaches. So if a case doesn't report a headache, they're going to ask them a little bit more. They're going to dig in a little bit more. You don't want that to happen. You want to have the same information collection procedures. Some studies will audio or videotape any interviews that happen to try to make sure that this is going to be conducted well and that they're going to do checks on their study personnel to make sure that data collection is consistent across their types of patients. You also can have a variety of issues that come up. Thinking about the reporting, we'll talk about bias at the end of today's lecture. But if your child was born and has a defect of some type, or maybe the child is born and starts having really horrible illness problems their second or third year of life, you're probably sitting up all night thinking about, what did I do during my pregnancy? What did I eat? What did my child eat? What were they exposed to? You're thinking of all of this stuff because you're not sleeping and you're reading different things. You're trying to figure out what caused this. If my child's perfectly normal, I'm probably not thinking about this as much. I'm not recording everything that they do and everything they ate and everything I can think about. So sometimes that recall bias, that different kind of perseveration that happens, is something that also impacts our information collection. We have to figure out ways to try to do that collection to minimize that bias that can happen. So our cases are, again, a well-defined population. I need standardized selection criteria as to what makes a case. Sometimes we do nested case control studies. We talk about that more in the book. Both groups are usually nested, both your cases and controls, inside a large cohort study. Where do I find cases? Well, sometimes in these cohort studies. Sometimes we have case registries. But again, who's going to get registered in there? You need to think about the potential bias and representativeness of the registry that you're using. We may look at admission records. So maybe you're trying to track down some hospital issue. You may look at admission records or pathology logs. There are problems with every method. The trick is to figure out which method is going to work best for the question that you have. The other trick is that cases tend to participate. Why? They want answers. It's pretty easy to sell a case to be in a case control study. They have a lot on the line. They want to know what's going on. There's no perfect control group, but you can try to get one. A lot of times you'll see matching on different variables that they think aren't involved to try to get your controls as similar to the cases as possible. You again need very standardized selection criteria, and you may have multiple control groups. Sometimes you'll see something called GenPop. It means general population. So they may pull samples from the general population to be a control group. You may pull people from a neighborhood. It might be the neighborhood they grew up in, that your cases grew up in, or the neighborhood your case lives in now. And that's a way to try to control for a lot of environmental factors that you may not be able to ask about. Families. Sometimes we'll do a twin study. One twin is affected with a disease, another twin is not, so we'll try to compare them to each other. And we may get hospital controls. People who were very sick at the same time but did not have the exact disease of interest. It's very costly to obtain controls, especially when you want to have multiple control groups. It's not uncommon to see a hospital and neighborhood control, for example. We're going to do adjustments of results during the analysis. Again, you want to use regression, not your chi-square tests. But also remember, controls, it's one thing if it's your twin sister, you might be more likely to participate. But otherwise, I live a busy life. Why do I want to be a control in your study? So it's hard to get controls. A lot of times you have to approach 8 to 10 controls before you get one that will participate. And that's being overly generous in my mind, but that's what the literature will tell you. So again, you'll see a lot of the same methods as you see for all the other studies. The validity of the measurement techniques are important, all that other stuff. You have all the biases that can happen, selection, observation, interviewer, etc. And then we try to summarize it in a 2x2 table. Please don't. But this is what those 2x2 tables will look like. You'll see presence or absence of characteristics, presence or absence of disease. The only reason I'm showing you all these letters, well, beyond the fact that it's in most epidemiology textbooks, is because those letters are used to try to show how you calculate odds ratios, relative risks, etc. So I wanted you to have the pictures. The analyses tend to be proportion of cases exposed compared to the proportion of controls exposed. A lot of times, while we use all these different tests, it should come down to regression. Again, when in doubt, use regression. Put a bunch of variables in the model. Positive attributes here. If you want to study the etiology of rare diseases, this is pretty much how you have to do it. If you want to study multiple factors simultaneously, again, these types of studies are good for that. You're collecting a ton of data to try to figure out what's different between groups. If you knew the answer, it'd be a lot easier, but you don't. So you're trying to collect a lot of information and see. It's actually not that time-consuming. It's less expensive than some studies. I'm not saying that it may not take you 10 years, but it might take you less than a year. The whole clause, if the assumptions are met, which is hard to know until after you're done and a lot more work has been done, the associations and risk estimates that you see in a really well-done case control study actually tend to be very consistent with other types of studies. So we've done case control, and then later on we're able to do additional randomized experiments and follow-on experiments. You actually, if you have a pretty representative sample in both your cases and controls, can get a very good estimate. But you're never going to know that until you've done the other studies. Negative attributes. You don't estimate incidence. Why is that? I set the number of cases in my trial. I don't know the incidence. I don't estimate prevalence for the exact same reason. The relative risk is indirectly measured, so I mentioned it there. But if you have a rare disease, so when you do case control studies, you can only technically directly measure the odds ratio, but you can get an estimate of the relative risk. We talk about that in the textbook. Bias is an issue, but bias is an issue in pretty much every study. You do methods to try to minimize your bias. It's hard to study a rare exposure. So remember, I can study rare diseases using this, but a rare exposure I need to use cohort studies for. And I probably want to sample on the exposure. So whatever's rare, you try to usually sample on if possible. Another negative attribute of these case control studies is that the temporal relationship is very hard to document. A lot of times people are remembering retrospectively what's happened, so it's hard to figure out the cause and the effect. But examples where this was done pretty well, this is a case control design back in the 1974, but I think it's really cool. I know. Actually, one reason I use all the older studies is then people don't sue me as much. But the other reason is some of these are just like good classic examples. This case control study identified the relationship between the exposure to a drug during mother's pregnancy with the occurrence of a rare tumor in the female offspring many years later. When I say many years later, I mean like they were 10 to 18 years old. How the heck do you figure that out? This is the type of stuff, though, that really well-done case control studies, this is the type of answer that you see. When you start seeing this very rare type of vaginal cancer show up in much younger folks than you would expect, you see a case series that gets published. And then you start finding all these different cases around the world and try to find matched controls. You go through all of the little girls' exposures. And then when you see nothing, then you start asking mom. And you start thinking, what's going on? Because also moms, when you got a 10-year-old, mom's there already. It's hard to remember. I mean, do you remember the drugs you took last week, let alone what you may have taken 10 to 20 years ago? But this actually held up over time. We have another one. This is one that Jerry Menikoff, who will be speaking to you, I think, in January, worked on. And so once I needed someone to cover for me teaching the epidemiology lecture, and Jerry's like, I've done an epi study. And he did a case control study, the risk factors for this post-operative endophthalmitis. I think I said that right. But basically, it was the link between this intraocular lens material and a serious infection following cataract surgery. So a lot of times you'll see if you're a medical student or you're a fellow, you start digging through medical records trying to look and do a study. And everyone had said, and he tells the story better than I will, but he basically said, like, the material couldn't matter. What are you talking about? Blah, blah, blah. But he kept looking. He's like, no, the type of lens that you use, the material in it, is very closely tied. And so he'd figured it out because of the manufacturer that was listed in the medical records for these different cataract surgeries. And ended up with a lot of material and actually a big change in manufacturing around the world for cataract surgery lenses. So now we're going to move on to cohort studies. Prospective or longitudinal cohort studies are your movies. All right? I tend to take observations concerning associations between some exposure or risk factor and subsequent development of disease. I might examine multiple outcomes for a single exposure. So what's this single exposure? So some folks online were talking about Liberia's Ebola survivors natural history study. I have to admit, I know next to nothing about this study. All right? So I could get the details wrong, in which case classmates, type it all out for me online. Because people started talking about it online today, so as I was in between meetings, I was trying to look up more and more information. But you could say in this case, and I'm not sure if it's a longitudinal study or not, I think it is, but I wasn't 100% sure from what I read online. But for example, I'm going to make it a longitudinal study for my example, whether it is or isn't. People have been exposed to the Ebola virus. They have survived. I don't know what's going to happen to them afterwards. We're starting to see problems with their vision. We don't know how long the virus is going to survive. Are they passing it on to their sexual partners, and for how long? There are lots of different outcomes that we may be interested in. When the World Trade Centers collapsed, CDC actually started a study of the people who were doing cleanup, and they said, all right, easy things we want to measure: respiratory distress and post-traumatic stress. But they measured a lot of other things about these people. They kept their blood samples, they asked them lots of questions, and they've been doing this over time. Why? Because we don't know what that exposure is going to cause in the end. So what these longitudinal cohort studies are good for is to try to measure a lot of different potential outcomes from a single exposure. They're also good to try to figure out the natural history of a population. So we did a study like this in people who are 65 and older in the United States. At first it was called the Cardiovascular Health Study. They enrolled people at certain sites around the United States that were on the Medicare rolls. And this study ran for 10 years, and they said, we want to know how Americans are aging in current society. What's happening? And that's what's useful about these longitudinal studies. You can directly calculate the incidence of disease for each exposure group in a study like this. You could do a lot of other really cool work too. So it's not necessarily always going to show you how to treat somebody, but it does give you an idea kind of what's the evidence and what's the underlying information. So here's your picture. There are two ways that this happens. Concurrent longitudinal studies. So today I define the population and their exposure status. You are exposed or not exposed. I'm going to follow you for a period of time. Let's say I follow you till 2025. So I'm going to follow you 10 years. I'm going to see, do you have the disease or not? If you were exposed to the Ebola virus, if you weren't exposed. If you're exposed to working on the World Trade Center sites, if you were not exposed to that. Or I do what's called a non-concurrent historical study. Today I see a group of people that have a disease or don't have a disease. And I try to say, what was your exposure status back in 1980? So I'm still defining a population by their exposure status, but it's in the past. How do you figure this out? Usually from work records. So a classic example is actually a nickel refinery study that I think was published by Leon Gordis in his epidemiology textbook, where they basically looked back at who worked at the nickel refinery and would have been exposed or not exposed to the certain toxic chemical. And then 50 years later, did they have cancer? You see this also, the Department of Defense has done several studies like this, where they look back at records from people who were there in World War II and what types of exposures they had. So let's go through, though, the concurrent prospective studies first. You identify a population that you want to survey. You define that. If you have risk factors in mind, which not all of these studies do, like cardiovascular health study, you were 65 or older on Medicare in the US, done. They had no other predefined risk factors, they just measured a ton of stuff. But if you have an idea of a particular risk factor exposure, again, you have to identify it, it has to be clear. You need to identify people with it and without it. Then you're going to follow them forward in time and compare the incidence rate of all these different diseases between the groups. If it's kind of retrospective, you need to define the population with the presence or absence of the exposure. And you've got to do that in an accurate, objective fashion. That's where those employment records come in. The data needs to be collected prospectively on the disease, however. Problem with retrospective studies is people are recalling information, and they may have very different recall. So we try to do this for some herbicide studies. That was a problem. We're like, well, do you really know if you were even exposed? Do you know what you were exposed to, how much exposure you had? It was very hard to try to get accurate information about exposure. But if you're doing these retrospective or non-concurrent studies, the idea is that what I'm surveying in the presence is the disease occurrence. Do you have the disease, yes or no? But again, I still have to define the incidence rates for those that are exposed or non-exposed. If you think you know what your exposure is, you need to make sure that your exposed and non-exposed are representative and well-defined, that your non-exposed are truly non-exposed. This can be a problem. The absence of exposure is probably going to be harder to define, to be honest with you. And the other trick is you have to assume that people maintained their non-exposure status throughout the study. So if you're going to follow them for 20 years and they suddenly get exposed in year 15, how is that going to impact your results? Something with traumatic brain injury issues happened. So there's a DoD study we'll discuss at the end of this, where they looked at people that had been diagnosed with traumatic brain injury during World War II. Then they were looking at their risk of Alzheimer's later in life. Well, there's a lot of things that can cause traumatic brain injury. If you're in a bad car wreck, you could actually have these similar types of injuries for what they were defining would have happened during World War II. So what do? Do you go through and ask people if they've had any of these following incidents happen? Or instead, do you go through and say, well, I'm just going to assume that they didn't? Which means that you probably have a lot more variability in those populations. All of the problems that I talk about can lead to incorrect estimation, incorrect interpretation. It doesn't always mean that the study is wrong. But it does mean as you're reading the study, and if you're the person that is analyzing it and writing those study reports, you need to say these are limitations that you have. You also hopefully can say, I'm going to try to collect other information and do what's called a sensitivity analysis to try to figure out kind of how wrong my data could be. How extreme do I have to be off before my study results change? And I tell the different story. All right, back to this. Your outcomes. What is disease? Death is easy to define. Everything else is hard. Or rather, not hard. It's subjective. So when does your cancer get diagnosed? That depends a lot on where you live, how much money you have, and if you do or don't have insurance and how good it is. It also depends on culture. There are some places I have been that, like for breast cancer, these women, it is bursting out of their breast, but they want to ignore it. You have to define the disease outcome, and you're going to probably have to build into your budget that you're screening for the disease in order to make sure that you have consistent outcome information. Your prospective or longitudinal cohort studies, the same criteria are applied to the exposed and the non-exposed groups. I keep saying this over and over because it's the truth. Definitions of disease have to be reliable and reproducible. Sometimes if you're doing that prospective study, the definition of disease may change during the course of your study. So kind of what the world thinks that definition is or how you go about checking to see and diagnosing a disease may change. In that case, you're going to have to update your protocol. And you're going to have to say, okay, here were my original disease definitions. What's my analysis with that? Here's the new guideline definitions that came out in the meantime. What's my analysis if I use those? You have to handle this. You also have to minimize your loss to follow-up because if you have a non-response rate greater than 20%, it's going to raise questions about the accuracy of your incidence rates. So if you have, they're looking for a certain number of folks that had had Ebola and a certain number of their close encounters in that natural history study. If they can't get those numbers, why? Why are people not signing up for their study? If they only do it in one specific hospital region, that's useful, but is it as useful and as generalizable if they moved across to other groups? So calculate the incidence for the study period. Again, you start to see all these chi-square tests, but really you should be using regression. The positive attributes of doing these types of studies, they're a lot more representative of cases than a case control study. Why? I'm picking up my incident cases now. You get a lot more natural history information when you do these studies. You watch the course of disease and all of the aftermath after diagnosis. It's actually a very beautiful type of study to do. The cohort studies I've worked on have probably been some of the more pleasurable experiences because you feel like you're able to actually give information to new patients that they didn't have before. Incidence rates are now available if you've done your job well, you have representative groups, and we can directly estimate the relative risk. So at the end of today's lecture, you'll have from slides 105 to 131. These are extra slides for you to read. There are actually a bunch of examples from my friend Steve Simon, and his website's listed on the slides so you can go dig in more into them. So that we're not going to discuss them tonight, but he talks a lot about how people basically more naturally interpret relative risk to odds ratios, and I think Paul Joachim will talk about that a little bit in his hypothesis testing. If you all want me to cover it more in the summary lecture, I can do that too. But it's a very tricky element to think about. So the positive attributes, there's a lot less bias when you use a cohort study compared to some other study types. However, they can be very biased also, don't get me wrong. You see a relationship to the exposure, and you can actually pick up a temporal relationship much easier prospectively than historically, but you can pick it up. You're more likely to. Rare exposure with frequent cases among the exposed show up beautifully here. The problematic attributes, you may need really, really, really long follow-up. It might take 50 years for dementia to show up. The free living population follow-up is expensive. It is hard to track down these people and to keep tracking them down every time you want to measure them. A large population is usually required in order to do these analyses and have reasonable power and reasonable belief that you actually have generalizable results. You need baseline data. If you don't get information about people close to their exposure time, you can run into a lot of problems. A rare disease really cannot be studied in this method. Again, you don't know if you have a big enough sample size to pick up enough people with the rare disease. But you can get rare exposures. So if you have a rare exposure, use a cohort study. If you just want to follow a bunch of people and see what's happening, do a cohort study. If you have a rare disease, do a case control study. You can see a lot of bias. So while it may be less biased than some things, when you see a lot of loss to follow-up, missed assessments, problems with how they do the assessments based on the exposure, you're going to see issues. Because again, here, oh, I know you had this disease, or I know you had this exposure, I'm more likely to poke at you. Ask you again, are you sure? So I mentioned some of these studies. There was the prospective cohort study that showed an early increase in the risk of lung cancer and heart disease mortality. And Dahl actually did this study. They had the original study of 54. Then they did a 50-year follow-up on these folks, a group of male doctors and their smoking habits. And they followed up in BMJ literally 50 years later. Then the medical records that I talked about, this is the study on that, looking at Alzheimer's disease and other dementias. And National Health Insurance Research Database. This is a study that came from one of the course participants a year or two ago, and he asked online, is enterovirus infection associated with an increased risk of childhood type 1 diabetes in Taiwan? So this is the question for you all to answer online. Tell me what the study design is when you read through this. And I think one of the PDFs you have is actually the write-up on this study. Here's the article if you want the link. The link was live at least on Friday when I checked it. Are there internal or external validity issues with this study? That's another question to ask. Then this is the study I mentioned on post-traumatic stress symptoms and asthma, looking after exposure to the World Center trade attacks. So not all cohorts, though, are exposure-based. Like I realized as I was going through the lecture, And I was reading it to myself, and I'm like, they're going to think everything is about exposure. But it's not. So this is a study also from one of your course participants on vegetarian diets and the incidence of cancer. So this is Adventist Health Study 2. They had close to 70,000 participants, and we're looking at incident cancer cases. I mentioned earlier a little bit about nested studies. So do realize that you might nest a case control or a case cohort study inside one of these cohort studies. So an example of a nested case control is from your prospective cohort. I've got all these stored samples, right? I've got baseline, follow-up data. I can now, a lot of times they'll wait maybe 6 months or a year. Then they'll take all newly occurring cases or a sample of those and compare those to matched or unmatched controls. What's nice? Well, I can go back and do that. I might be at year 5 or year 10 of my cohort study. I followed those controls for a while. I know they didn't become a case. They're still non-cases. So I know they're a control. I know they don't have disease. This is really efficient, especially if you have expensive or difficult lab assays you want to run. You store everything, and then you're only going to run the assays on a sample of folks. And it might be nice because you can see, do I have enough cases in the cohort in order to actually even bother to run the case control study? You also don't have as many selection and data collection issues. You can still have them, but there may be fewer. In a case cohort study, you're selecting from the cohort all incident cases and comparing to a random subset of non-cases. And you can read about this more in the textbook. In kind of our advanced chapter, we go a little bit more into this. These are pretty difficult to analyze. So in general, you've got a lot of different types of observational studies. They fall into kind of these key categories: a case reports, a case series, cross-sectional surveys, national health interview surveys, another common survey that you see data from, at least inside the United States. Case control studies, so these are groups with or without the disease outcome, and then you determine who's exposed to different risk factors. Or cohort studies, where you follow a group for a while, or you're going to choose people with or without an exposure status. So now let's talk a little bit about bias. Some folks are going to talk to you about 20 different types of bias. I don't care about the labels. Other people do. So when you take their exams, care about the labels. As a statistician, all I care about is there's error. And so there's a true value and a value you collected in the study, and they're not the same. That's bias. I don't particularly care if it's selection bias or interviewer bias or anybody else's bias. There's a problem because I'm not getting the correct information. But like I said, a lot of other people care about the exact names. I don't. Bias is bias. But I'll go through a few of your common names that you're going to hear in the next few lectures. Selection bias, sometimes called prevalence incidence bias. If you're exposed or impacted early, I might miss you. Why? The fatal episode. So if I go out to survey who has Ebola right now, if you already died, you don't have Ebola right now, you're dead. Maybe I didn't know yet that it was Ebola. So even if I ask, people don't know what to call it. Transient episodes might be missed. The silent cases. If you had a silent stroke, nobody knows about it until you go in for something else and they figure it out. Cases where the evidence of exposure disappears with disease onset happens a lot more frequently than you might expect. Non-respondent bias. People were unwilling or unable to respond to your kind survey. You don't know about them. And what you also don't know is if you would have had different exposures and outcomes in the people who were responders and non-responders. So sometimes you get this diagnostic suspicion, exposure suspicion, recall bias, family information bias. And this can go in a variety of different ways. Again, let's say in your family that this disease is somewhat prominent, maybe has a genetic aspect. So if you come from a family that has Huntington's disease in it, you're probably going to be looking for and thinking about some of the early ways that you would diagnose Huntington's disease beyond the genetic test. At the same time, I had an aunt for years. I thought that she had died from a stroke in her 40s. That's what my dad told me. So when I started having severe headaches in my 30s, that was in my family history. My doctor did a full workup on me. Then I found out that my aunt had killed herself. She committed suicide, but that was considered so taboo in my father's family, they lied to everybody about it. So sometimes we talk about our histories, and sometimes we don't tell the truth about our histories. Those can cause a lot of bias. So in conclusion, observational studies are really useful. They could be your only alternative. I love randomization. I really love randomization. I have come up with randomized trials when people told me there was no way to randomize, and we figured out a way to randomize that study, and it worked. But sometimes it is not an alternative. I cannot randomize smoking in humans. I cannot randomize people to long-term antiretroviral therapy for HIV. It may be unethical, or maybe they're just not going to stick to it for that long. There are going to be too many changes. What am I going to interpret at the end of the day? If you cannot interpret the randomized study, you have to really think about what your other options are. Sometimes my question is not randomizable. Cardiovascular health study, what happens in free-living people? You don't randomize that study. And sometimes people fall back and they say, well, it's cheaper and faster than a trial to do an observational study. Well, that's true too. But cheaper and faster may not be better. So you have to balance kind of what you need. Yeah, everyone's laughing about that. But seriously, we have gone off the rails on studies. And I will also say that for randomized studies. You have to do a good job. If you can't do a good job, just don't do the study. Observational studies do not always agree with themselves, and they do not always agree with our randomized research. Hormone replacement therapy is a very long example of that. You can read about it extensively, or we can talk about it. But there is also many times publication bias, and you have to take that into account. Incorrect analysis of observational studies is also a big issue, especially if you are looking back in time. Sometimes you have different populations. The population that was in the Women's Health Initiative was very different than the population typically taking hormone replacement therapy. That has to be taken into account. You also have to think about what are the populations for your epi studies and your randomized trial, and what's your general population. Observational studies will find a weaker degree of connection. That's what they're there for. You can correct for what you know, but not for what you don't know. But again, if it's unethical to do the randomized study, you're just going to have to do a really good job of your observational one. You're going to have all the design issues, placebo effects, investigator and participant biases, unblinded treatment assignment and assessments, because that's essentially what free living is, right? It's basically an open label experiment. You got regression to the mean. You're going to see natural reduction in disease activity over time. Same problem in observational studies. Got causation versus association. Your causation can only come when you have some randomized experimental study or clinical trial involved. Your association can come from a lot of different types of studies, randomized and non-randomized. So, what do you need to do? Measure everything you can, build and investigate a lot of different models, and be honest about that. Don't just publish the one that looks good. Test the models on a lot of different data. Try to replicate it in a lot of different data sets. Try propensity scores. Try tons of different methods. Have a reason for your methods, but you're trying to generate hypotheses from this. There are going to be a lot of problems, but your biggest mistake that I've seen is designing a randomized trial because they're like, that's the best evidence, when you really need an observational study. Look at the alternatives. Think about the predecessors, your follow-up studies. Think about population assessment. Do you have the information you need to do a randomized study? Granted, You should also be willing to do creative randomized studies if that's what you need. Here's some cases where I've been able to randomize, but a lot of times you can't. We can discuss those more online if you want, but you've got to defend your non-randomized studies. Randomization is your research norm, so you need to admit your limitations and defend why you need to and how you're going to minimize the problems that can come up. But don't forget that observation is your first step for any research. Don't be afraid to look. If you want more information on publishing guidelines, they're published in many different ways. Epi-studies have their own version of Consort. You've got a lot of self-guided course information here from the CDC. And thank you, and if you have any additional questions, please go ahead and submit those online. We have some great lectures tomorrow night, and I hope to see you all in the coming weeks. Thanks a lot.